So in this recording, we want to address the nature of the information in DNA, and that is the genetic code. What is genetic information? And the cool thing about this, which is pretty profound when you think about it, is that this is true for all life. So basically, everything that we've ever seen that's alive follows the same genetic code, which is a strong piece of evidence that, because this is so fundamental to how a cell works, that all life originated probably from a single event. All right. So the genetic information is a triplicate code, and that is that there are 3 nucleotides in DNA that basically encode for one amino acid. Right. So each of these triplets is called a codon, and the codons then encode for the 20 different amino acids that are used by living things to make their proteins. For example, an AGT is going to make the amino acid serine when it is read from the DNA. All right. So a handy chart that you're going to see again and again, and we're going to practice using, is here that shows you all the possible combinations of groups of 3 of our 4 bases. Now this one happens to be an mRNA chart. So the only difference between this and a DNA genetic code chart is that all the U's are T's. So here we see U's because we're talking about RNA. In the DNA one, they would all be T's, but it's in essence the same thing. Now the way this works is on the left-hand side, you see it says first mRNA base. So remember, we're going to have 3 bases in a codon. So the first mRNA base, you pick one, U, C, A, or G. Let's pick A for an example, and then you would read the second base. So if we had the triplicate, the third code, if the 3-letter code that we were working with was AGU, we would pick A from the left side. Now we're in that row. And then when we go to G across the top, so we'd be on the far right column. So where those 2 meet is a square where you see 4 combinations. A is first, G is second. And then our third is U. And so we see AGU, that's our third on the right side. Within that row, UCAG again, you see AGU equals SER, serine. All right? So that's how you use one of these charts. You'll also notice there are 4 special amino acids. AUG, or ATG in the DNA, is a methionine always, and it is the start codon. It's always going to be the first codon, and it's going to allow the molecular machinery to orient itself to where a gene starts. Then there are 3 what are called stop codons. They are UAA, UAG, and UGA. And to remember those, and this is why I use the RNA chart for this, because they're easier to remember. When you say you are awful, that is UAA, stop codon. You are gross, UAG, stop codon. You go away, stop codon. So UAA, UAG, UGA are the 3 stop codons, and you should commit those to memory. All right, so once I have that information, knowing that the code is a triplicate-based code, I can look at this and understand it a little bit better. So again, we've got our DNA template here. It is a double-stranded molecule. There's our gene. We zoom in on it. You can see we have 2 strands of DNA. Now, one of those strands encodes for the gene. And we're going to use the opposite strand then to build an mRNA molecule that looks like the sequence that we need. So in this case, we can see that the bottom strand is oriented 5' to 3' in our double-stranded DNA, and that is the gene in essence. So this gene would start with a TGG, TTT, GGC, TCA. We always read genes 5' to 3' because that's how a ribosome reads genes, 5' to 3'. So the 5' most codon will be the first amino acid in the protein. So the opposite strand then to the gene is called the nonsense strand, and we'll get to that in a second. But that's going to be the template then that we build the mRNA from. So you can see through the process of transcription, which we'll get into details a bit later, you can make an mRNA molecule, now 5' to 3', and then you can basically start to break those down 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. And then translation gives you an amino acid for each of those codons. Granted, this one doesn't start with methionine, but it's just an example. Most proteins will start with methionine. So let's go back to that idea of template and non-template strands. So if I have a piece of DNA, if I were to isolate your DNA, and I quote-unquote read the sequence, well, I only need any one strand to get both strands, right? Because I know the complements of every A, T, G, or C. But only one of those strands will give me an amino acid sequence that means something to the cell. And the template strand is going to be used for ordering the sequence of nucleotides in the RNA transcript. So when I read that one, it's going to be nonsense. We call it the minus strand, the nonsense strand, the antisense strand, the non-coding strand. But it is the template then for building the mRNA. So the non-template strand, the one that looks just like the mRNA that's going to be built, we call that the sense strand or the plus strand or the coding strand. So that one, where I to read it, I can actually get then the amino acids because I know the genetic code that will comprise the protein. So again, if we have our double-stranded DNA and I pull it apart, the sense strand is going to be the top strand, 5' to 3'. In this case, the bottom strand is the nonsense strand. But it's the nonsense strand that I'm going to use as a template to build an exact copy of the sense strand, and that in essence is going to be my mRNA that is going to go out to make my protein. All right. So codons are read 5' to 3'. There are 64 specific combinations of groups of 3 of 4 letters. All right. So 61 of those are going to specify amino acids, as we've already seen, and 3 are reserved for stop codons. So the problem is we've got 64 codons, but we only have 20 amino acids that we really need. And so what we have is a code that is considered degenerate. So when you have redundancy in the code, if you're a code person, I guess the name for that is a degenerate code. So what you have to do is be careful that the right reading frame is established, that you start your gene at the right spot so that you don't read just any group of 3. All right? And we'll come back to that idea again and again. I just want to introduce it now. So understand the genetic code, that we have groups of 3 that we call codons, and each one is known to encode for a specific amino acid.